So thank you for coming. Before I get started, I just want to tell everybody there's a power at my house tonight, which is walking distance from the university, 1756 feet circle, to anyone who's like to know afterwards. And before you get it from Roxanne. So that was sort of around 6 or so for a little celebration. So Roxanne came to University of Western Fairbanks from UAA. Before that, I discovered that she placed 4th in California State High School Science Fair.
Oh no. Placed 4th.
Interestingly, here's when it should be a different year than it does on the state webpage.
Oh.
So I don't know if that's true. After that, she went to UCSC and worked in Dan Costa's lab, where I did postdoc, so we had some. I don't think we quite overlapped. I think you left a few years. I got there. I left a few years before you got there. And you published and you're doing undergraduate 4 papers, something like that. Hardly any for undergraduate. Then she went to UAA and worked with Jen Burns on whale seals in the Antarctic. Was awarded while she was there an NSF-GRF, and then pursued a PhD where she came to UAF. And so, and after this, after she's already lined up NSF postdoc back in Santa Cruz, so she's working pretty hard and really making waves. So we'll let Roxanne tell you about her work in the Antarctic.
So thank you all for coming today. I am really excited to talk to you about the work that I've been doing over the last 5 or so years on the behavior and physiology of Weddell seals in the Antarctic. Before I start, I just want to thank my committee members, Diane O'Brien, Brian Barnes, Ward Testa, and Greg and Jen for contributing intellectually to this work. So I'll begin today by talking about how life history theory shapes our understanding of the natural world. Life history theory provides a nice framework for understanding how animals trade off current performance metrics, such as reproductive success, with future performance metrics. And of course, these trade-offs result from the limited resources that are available to animals that must be allocated to one life history event or the next. Now, typically, we think of environment as driving that resource availability, and so environment really strongly affects those trade-offs. The timing or phenology of life history events, I think, fits nicely within this framework. So here I've put a conceptual frequency distribution of offspring birth dates for a mammal species, like Weddell seals. Animals that give birth early in the season tend to be of lower maternal mass, but, for example, produce offspring that have greater survival during the offspring dependence period. But because they're older and more experienced and fatter during weaning, they tend to survive more frequently. Whereas conversely, animals that give birth late in the season tend to be born to moms with greater mass because those moms have had more time to accumulate that mass over the season. And so the offspring tend to survive more during the dependence period, but because they're younger and more experienced at the weaning period, they tend to survive less. And so this trade-off between, for example, pre-weaning and post-weaning survival, I think, is an example of how selection may stabilize the phenology of birth dates. In this specific example, environmental conditions are driving the window, the timing window, within which those life history events can be performed. Particularly in polar regions, these drivers can be really seasonal. So, for example, food availability, temperature, light, and predator abundance can all really strongly influence when life history events can be carried out. In other words, we can say that these life history events are under strong selective control. I think one of the coolest examples of this is primary productivity across seasons for different regions. So here I plotted polar regions. You can see there's a really extremely high but short-lived peak in productivity during the summer. And then resource availability or primary productivity is lower throughout the rest of the year. Whereas tropical regions, for instance, are more homogeneous, kind of in the mid-range of primary productivity. So this seasonality leads to geographical variation in life history traits or life history phenology. I think one of the most impressive outcomes of this is the migrations of many bird and mammal species. So here you can see as the snow begins to retreat from the northern hemisphere, there's this really impressive pulse of animal migration to the poles as animals try to take advantage of those resources. And then after the summer, will tend to leave to avoid the winters. So this is a really common strategy in the animal kingdom. But another strategy is animals that reside in these really harsh polar regions year-round. Today, I'll be talking to you about some of these polar residents and how they manage to endure the harsh winters and conversely take advantage of the productive and warm summers. So as a result of the extreme seasonality in polar regions, these animals undergo their life history events in quick succession. They give birth, they raise their offspring, they wean those offspring, they feed, they breed, they become pregnant, and they replace their fur or feathers during the molting process, all in quick succession in the span of a few months or just a little bit more. And that, to me, begs the question of how are these events linked in time, and how is energy allocated to these various life history events? So for my doctoral research, I explored this broad question by studying the most southern breeding mammal, the Weddell seal. The resulting dissertation consists of 4 chapters, 3 data chapters and a review paper. And just to give you an idea of what those chapters are, I have a diving behavior chapter. In my second chapter, review molt strategies in birds and mammals. And then in the 3rd chapter, I look at the timing of molt in Weddell seals. And in the 4th chapter, I validate a method for estimating mass in this species. So today, I'm going to integrate all of these findings into a single narrative, and I'll point out each of the chapters as I go along. So I'll talk about 3 kind of specific questions that I addressed in my dissertation. First, I wanted to identify how the phenology of pupping, foraging, and molting are linked during the summer in Weddell seals. Second, I wanted to evaluate how these events influence the probability of successfully producing a pup in the following year. And third, I wanted to evaluate how all of these events are affected by ice dynamics. So I'll go through a bit of fieldwork methods first. Our study location was located at McMurdo Station, Antarctica, at 77 degrees south latitude. And I don't have time to talk much about the fieldwork today, but I just want to say that a huge amount of time and energy went into this, both from members of our field team, but also from the contractors in the United States Antarctic program that kept us warm and safe. So we did this work over the span of 4 years, from 2013 to 2017, and we were traveling down to McMurdo during the austral summer, which in the southern hemisphere is November through February. We are not advancing. All right. So thanks to that logistic support, along with the high survival rates of Weddell seals, as well as their high philopatry, there's a 55-year demographic study on this species. So essentially, almost every pup at birth is tagged using these unique flipper tags you can see. And they're individually identified at birth, such that most of the population of the 2,000 animals, we know age, sex, birth dates, colony attendance patterns, reproductive success, reproductive timing, and survival. So there's a huge amount of information that we can use to really address novel questions. I want to thank our collaborators at Montana State who did all of this work and were willing to share the data with us. Okay, so I'll introduce you to the Weddell seal annual cycle. It's centered on the warmest part of the year in the southern hemisphere, which you can see in January, represented by that sun. So Weddell seals give birth to pups in October. They nurse those pups for about 45 days. And then after a short period of breeding and some summer feeding, they undergo their hair replacement process in about January and February. And then following that becomes the energetically expensive part of gestation, and the winter follows that. So for the first part of the talk, I'm going to walk through these life history events in sequence, starting with pup birth. There's quite a bit of variability in pupping success in Weddell seals. So for sexually mature females, we can categorize them into one of 3 categories. The first is puppers. Those are animals that attend the pupping colonies and are repeatedly sighted with a dependent pup. That's about 2/3 of animals. There's a second category that I'll call skippers, and those are animals that do attend the pupping colonies but do not produce a pup in a given year, despite having previously produced a pup. That's about 1/3 of animals. And then there's a third category that's called missing. Those are animals that are not seen at the pupping colonies and are assumed to have not produced a pup because we know with relatively high confidence that these animals are not producing pups outside of this particular system. And those are animals of two categories. They're ones that temporarily emigrated, so they may have left and then they'll come back the following year. That's a small subset of animals. And also animals that may be deceased, so they're just never seen again. But because it's difficult for us to disentangle deceased from temporary emigrants, I combine both of those into a single category that's called missing. So for the sexually mature females, we classify them as either puppers, skippers, or missing animals. So the question then becomes, why don't adult female Weddell seals produce pups every year? We know from measuring their mass during both the pup birth period and the pup weaning period that puppers are losing about 40% of their body mass over the lactation period, whereas skippers are likely maintaining that mass because they don't have to deal with the energetic costs associated with lactation. We also know that skippers have a 10% increase in pupping probability the following year. So given these obvious costs of reproduction, we hypothesized that Weddell seals must have a strategy for regaining mass after that lactation period and in preparation for the molt that's coming up and for gestation as well. And that brings me to the next life history event, which is the summer feeding period. So typically to study seal behavior, we glue tags to the animal's fur. But because Weddell seals molt right after this, the tags would just fall off. From gluing instruments on Weddell seals, we know that they're really great divers. They can hold their breath for an hour and a half and dive down to 900 meters, which is the height of a 300-story building. So they're really incredible animals, but we don't know what they do during the summer after that lactation period and before they begin molting. Fortunately, with the advancement of telemetry devices that are now very small, we can attach those devices to the flippers of animals, so it circumvents the problem of hair loss during the molt. And that's what we did for the first chapter of my dissertation I'll be talking about. We put these miniaturized hand-up recorders in the flippers of 95 adult females. These tags are about 20 grams, and they essentially record the animal's depth in the water column every 6 seconds. We were able to get 59 tags back, and that gave us data for about 230,000 seal dives. So to give you an idea of what this data looked like, here I'll plot depth on the y-axis, from 0 at the surface down to a couple hundred meters, and then time on the x-axis. So for a 4-minute dive, you'll see a blue line appear. The animal dives down, and then chases fish up and down in the water column, then comes up for a breath of air. So at the bottom phase of the dive, where those vertical excursions are, we call those wiggles. And we can calculate, using a custom program in MATLAB, the number of wiggles, along with the start and end of the dive time, and the start and end of the bottom phase. So by doing that, we can essentially calculate the duration of each of the dives and automatically identify each dive from the next dive. So the first thing I wanted to do was calculate the percent of time these animals are spending diving. So I summed the dive durations and divided that by the deployment length that the tag was attached to the animal. And I found that skippers spend about 30% of the time diving, whereas puppers spend about 9% more time diving than skippers do. So these data provide supporting evidence for our hypothesis that the foraging period following lactation is important for the puppers to recuperate mass after lactation. Now, if I zoom out to 2 months of diving, again plotting depth across time, you can see that each dive essentially gets vertically compressed into a single line. And this is how Weddell seals dive. They do groups of dives that are called bouts, and then they come up and rest for a bit at the surface to sort of physiologically recover. So according to optimal foraging theory, these animals can maximize energetic efficiency by diving as shallow as they can in the water column and still feeding on fishes. So the assumption is that they're not going to pass up huge groups of fishes in the water column, which essentially means that the bottom of this dive record is sort of a loose approximation for the shallowest distribution that the fish they're consuming are. When we went into the study, we had no reason to believe that there would be any sort of seasonal pattern in this dive depth. We know that they eat these fishes that are found at a couple hundred meters depth, and we had no reason to believe that there would be any sort of seasonal change in the distribution of those fishes. So that was our null hypothesis. But when we started getting the data back, we saw this really clear pattern that, for example, this first seal was following, where the seal was diving deep to about 400 meters at the beginning of summer, and then diving shallower to about 100 meters in the middle of summer, and then diving deep again. And this is weird. This is not normally seen in seals. I'd never seen this before. We got back the second tag, and the third tag, and the fourth tag, and we realized that this was more the rule than the exception. So we wanted to kind of dig a little bit deeper into this and try to figure out what was going on here. In order to look further into this pattern, for each individual seal, so this is one seal, we plotted depth against time, but this time each dive is represented as a dot, so it's the maximum depth of each of those dives. So you can sort of see that deep shallow deep pattern. And we quantified the 95th quantile of dive depth for each seal for each day. The reason we did that was because we weren't interested in the under ice dives and we weren't interested in the benthic dives. We're interested in how the pelagic dive depth changed across the summer. So that 95th quantile was taken for each seal for each day, and then we took an average and standard deviation of all those values across all the seals. And again, we saw very clearly this deep, shallow, deep pattern. Here I put a box around the shallowest 3 weeks of diving for reference. So this pattern was really compelling to us, but we wanted to ensure that, that the seals were actually feeding at the bottom of their dives. So in addition to deploying those time depth recorders, we put out a subset of jaw accelerometers. So the way these tags work, they measure raw acceleration, just like a Fitbit. And so every time the seals open their mouth, it records a big surge in acceleration. So you can see that in the plot in the top right. Each time the seal opens its mouth, it's these big surges in acceleration. We have a custom algorithm that can essentially identify each one of those jaw motion events, which is an assumed prey capture attempt. And we can overlay those onto the depth record, which is what you see in that bottom panel. So with these data, we know where in the water column and when in the dive the animals are presumably feeding. And as you can see, most of those occurred during the bottom phase of the dive, like I said before, with the vertical excursions or the wiggles. So we looked at the association between wiggles and jaw motion events, which was strong and positively correlated, and also between wiggles and masking, which again were strongly positively correlated. So we use these wiggles as metrics of presumed foraging success. And then we can look at those across the summer to assess whether there's higher foraging success during that shallow period of diving. And we found that there was. So there's our square reference box there. You can see this is wiggles controlled for bottom time as a function of time. And that increases across the summer, is highest during that shallow period, and then begins to decrease. So it seems that not only are these seals able to dive shallower during this period of shallow diving, but they're able to have higher presumed foraging success as well. So I began to dig through the literature and try to figure out what was happening here, and the pieces of the puzzle began to fall together. I realized that the dive data that we had was giving me interesting insights into the ecosystem. So I'll show you what I mean using this conceptual figure. Again, this is depth across time. We know the seals are diving deep, then shallow, then deep. That shallow period aligns really nicely with when the ice breaks out. And we know that the ice breakout triggers the phytoplankton bloom every summer. And that increases the chlorophyll concentration in the upper 50 meters of the water column by about 20-fold. We know the food web in our system is relatively simple, so the seals are eating, this is very simplified, but the seals eat silverfish, the silverfish eat zooplankton, and the zooplankton eat the phytoplankton. We know that the silverfish and krill are usually distributed at a couple hundred meters deep before and after the phytoplankton bloom. But we know that their reproductive period aligns with the phytoplankton bloom, and that they become shallower during that time as the eggs are positively buoyant. During this time, we also know that seabirds and penguins, which can't dive very deep, have been found to feed on deep dwelling fishes. And so it seems like maybe both birds and mammals are taking advantage of some sort of vertical distribution shift of the fish and krill in this system during the phytoplankton bloom. Now, if we look at a timeline of what I've just showed you, instead of looking at it in space, I've already showed you the Weddell seal annual cycle, right? So there's this period of offspring dependence that ends in about December, and then they become independent here. And that period of independence aligns nicely with when the silverfish and krill are spawning, and also when the phytoplankton bloom occurs. Now, if I add on here emperor penguin period of independence and Antarctic minke whale period of independence, it seems like the weaning periods for all these animals are sort of aligning with that phytoplankton bloom. And so I think that this presumed period of shallow prey availability that I'm proposing could be a really interesting selection pressure on the reproductive phenology, as well as the spatial vertical and horizontal distributions of both birds and mammals in this Antarctic system. Okay, so after the summer feeding period that I just talked about, the Weddell seals begin their annual molting process. But before we can characterize the links between life history events, we first need to understand each life history event in isolation. The molt, I would argue, is one of the most poorly understood things that mammals do, but I think it's one of the most important. So if we take a step back, we can really appreciate the importance of molt in terms of maintaining fur and feather function. We know that fur and feathers allow animals to stay warm and to blend in with their environments, to attract mates, and to fly. But as the feathers and fur degrade over time, they lose their function. And so each year during the annual molt, animals can essentially replace those fur feathers to ensure that those functions are maintained. In the species that have been studied, a wide variety of molting strategies have been identified. And these are driven by selection pressures on their function, either via natural selection from an animal's habitat or niche, or from sexual selection via an animal's social dynamics. Now, when I started reviewing the molt, I realized that, as far as I can tell, there are no mammals that use hair color for mate choice dynamics. And so it seems that at least for mammals, the predominant requirement for their hair is to blend in with their environment rather than to attract mates. And so there's kind of an interesting difference between birds and mammals, in that birds tend to use more social dynamics for that plumage selection, whereas mammals tend to follow more the natural selection pathway. So we reviewed the contribution of these various pressures to birds and mammals and their molting strategies, and I'm happy to say that paper came out last week, and it's available open access if you'd like to see it. But I'll give you a sneak preview of what I think is one of the more exciting components of it, which is sort of this decision tree for how different molting strategies arise. So some animals exhibit a continuous shedding pattern. I think that this is sort of predominantly domesticated dogs, especially breeds that aren't in Alaska. But they do, instead of having an annual molt that's during one time of the year, they'll tend to shed throughout the year. On the other end of the spectrum is animals that do have seasonality in resources, do have seasonality in insulation requirements, and do have seasonality in their camouflage requirements. So like animals that live in snowy environments tend to have a complete biannual molt strategy, where, for example, with rock ptarmigan and with snowshoe hares, they're molting into a brown plumage or pelage before the summer, and then molting a second time that year into white in order to blend in with the snow. To go back to the decision tree, we know that if animals have seasonality in resources and insulation but not in camouflage, an incomplete biannual molt can come about, where, for example, with elk that don't live in really snowy environments, they can grow a thicker pelage before the winter and then shed into a thinner one after the winter. And a similar incomplete biannual molt strategy can come about just in birds to get into that breeding plumage. Again, for example, with peacocks, molting into a colorful breeding plumage for the breeding season and then molting out of it to enable crypsis throughout the rest of the year. And finally, with very limited seasonality, we see that many species also instead have an annual molt. And we know that seals and sea lions fall into this category. So getting back to Weddell seals, we know that they have a single molt per year, but to fully understand it, we need to know how long it takes, when it starts, and also how reproduction affects it. Because of their site fidelity, these animals are really easy to study during the annual molt. So for another chapter of my dissertation, I quantified Weddell seal molt duration and timing. So to do this, we visited seal colonies every few days. This is an example of a sighting interaction where we document the tag number and the molt status of an animal. You see, they're pretty approachable. And we did this for 4,000 individuals with over 9,000 sightings. So I want to particularly thank Amy, who helped enter every single one of those data points. It was really great to have that large set of data to answer these questions. So in terms of documenting molt progress, Weddell seals molt in a very characteristic pattern. At molt code 0, they have not started their molt yet, so they're really scuzzy brown color. And then as they start their molt, you can see their head and a stripe along their back becomes molted, so you can see that new fur showing through. And then that dorsal stripe widens, and that's called a molt code 2. Molt code 3 is when they're almost finished molting and they just have a bit of old fur on their sides. And then molt code 4 is when they've completely finished their molt. So at each of those sightings, we can record the molt code that they're in, and then we can put all of those molt codes across time for each seal to start to get an idea of how long the molt takes. The way we do that is by finding the last time they were seen unmolted and the first time when they were seen having started their molt. We calculate the difference between those two, and those become the durations for each of the molt stages. And that's important because, for example, if we first see an animal in molt code 2, if we know the duration of molt code 1, we can back calculate a start date for that animal. But also, by summing each of those 3 components, we get the entire molt duration. So we did that and found that the Weddell seal molt lasts about 1 month. Now, relative to other seal and sea lion species that has been studied in, the molt takes a little bit longer than in the monk seals and elephant seals, which are true capital breeders, in that they finance breeding and molting via onboard energy stores. Whereas the otariids, like New Zealand sea lions and northern fur seals, tend to feed during molting and lactation, and so their life history events typically take a bit longer. So Weddell seals, along with harbor seals, are sort of in an intermediate category, where they do a bit of feeding during these life history events, but the life history events are on the shorter side. Okay, so now that we've placed Weddell seal duration in the context of other species, we can look at the phenology. So in repeating these analytical procedures for many seals, we can begin to see that some seals start their molt early and some seals start their molt late. So looking into how that variation in molt phenology is split across categories gives some really interesting insight. So here I've plotted the molt start dates for skippers. So we can see that they start around January 1st, and there's a wide spread of about 50 days for when they're starting their annual molt. But if I add puppers, you can see that they start significantly later, about 20 days later than the skippers do. And then the female immatures, so females that have never given birth before, and males fall intermediate to those two. Now I wanted to dig a bit deeper into this and just take the puppers and look at whether pupping phenology influenced molt phenology. And so the way I did that is by calculating the distribution of pup birth dates. This is similar to what I showed you in the first couple slides. We had early puppers that gave birth in the first 25 percentile of birth dates, late puppers that gave birth in the last 25 percentile, and then mid puppers, which fell in between. Similarly, for the molt start dates, we calculated early molters, late molters, and mid molters. And I'll use these color schemes for the next couple slides. So here's a schematic of what this chapter looks like. So you can see we have these skip puppers during the pupping season in gray. We have early, mid, and late puppers in progressively darker shades of red. And so each of the animals from that pupping category then transitions into a molting category a couple months later. They transition into either early, mid, or late molter. So if there was no influence of pupping timing on molt timing, we would expect that these animals would sort of randomly distribute into the molting categories. And what that would look like in terms of results is here we have the proportion of individuals that are comprised of the different pupping categories. In the null hypothesis, we would expect those would be evenly distributed. But what we saw instead was that there was a strong influence of reproductive success and phenology on molt. So the early molting category was really heavily represented by skippers, 28% more than expected than by random chance, whereas the successful puppers comprised a much smaller proportion. Whereas during the late molting category, we saw that there were hardly any skippers, but that early puppers, for example, contributed 9% more than expected by random chance, mid puppers contributed 12% more than by random chance, and late puppers contributed 20% more than by random chance. So it seems like by skipping reproduction, would also in the molt much earlier relative to those that do produce pups in a given year. And also it seems like pupping timing is related to molt's timing. This relationship between reproduction and molt is likely driven by reproductive hormones, such as estrogen and prolactin, that are elevated during the lactation period and likely preclude the molt start. So prior to this study, the effect of successful reproduction on molt timing had been found in 7 species, and so we can add to this with a pretty large sample size, which is exciting. But ours is the first study that I'm aware of that actually links late reproductive timing to late molting. And I think that just stems from the difficulty of quantifying both of these metrics for the same individual in the same season. Okay, so now that we've established there are links between pupping, molting, and foraging, I want to move on to talking about how these events influence pup production in the following year. So I brought back this schematic. We're now looking at the transitions between molt timing in year 1 across the winter period to the pupping season. And animals can either be missing, which is either temporarily emigrated or deceased, or they can be skippers or successful puppers. So I'm going to fill in this empty matrix with transition probabilities from molting categories on the left to pupping categories on the right. And for simplicity, I'm going to use arrows. So a green upwards arrow means there's a higher probability than expected by random chance that that transition will occur. A red downwards arrow means there's lower probability than expected by random chance, and a 0 means there's no difference. I just want to point out that I used Markov simulations on a multinomial draw to calculate whether or not these were significant. So mid-molters had no significant difference from random chance of going to any of the categories, but we saw that for Weddell seals that are early molters, they had a lower probability of going missing and a higher probability of attending the colony as skippers, whereas late molters had exactly the opposite probability. They were more likely to go missing and less likely to attend the colonies as skippers. So notice all the zeros in the pupping column. It appears that molt timing is more related to colony attendance than it is to the probability of reproduction in the following year. So if we take a step back and discuss what we know about attendance versus emigrants in terms of Weddell seals, like everything in the Weddell seal annual cycle, this is a trade-off, right? So animals that attend at the colony are given breeding opportunities and predator avoidance opportunities, but in terms of cost, they can face more conspecific conflict or food competition. We know that in immature Weddell seals, more sea ice extent leads to higher rates of temporary immigration. And that's most likely because of the links between primary productivity, foraging success, and body condition, where it's been proposed that there's some sort of threshold body condition for colony attendance. And so if the ice dynamics are thrown off and there's lower foraging success, then animals may not be able to attend the colony. So we hypothesized that because of these links, colony attendance may be related to ice dynamics in a given year. And so that brought about my third research question, which is how are these linked life history events affected by ice dynamics? We were really lucky in that this study spanned 4 years with a lot of variation in the ice breakout phenology. And so we had a really unique opportunity to explicitly address how ice dynamics might influence these life history events. So I'll show you again a map of our study site. The red star represents McMurdo Station. And as the summer begins and the air temperature warms, we see that the ice essentially breaks out from the northeast corner down to the southwest corner and eventually reaches station. So I'll show you a satellite image of what the ice looked like in early January of 2015. So you can see the black is water, the white is ice. This is where the ice edge is. So in early January, it's within about 20 kilometers. Now, if I overlay the satellite image from the same calendar date, but 1 year later, in 2016, we see that the ice edge was almost 80 kilometers further. So the ice took a lot longer to break out in that second year, and the difference between the 2 years in ice breakout date was about 30 days. So we can take those 2 years and look at how ice dynamics may have affected the patterns that I've already talked about today. First, we can look at that deep, shallow, deep diving pattern. So what you're seeing here is depth across time. Each of the dots is one of those 95th quantile points, and then we have a GAM fitted to those data. So this is the deep, shallow, deep period for 2015, the earlier ice breakout year. And now I'll add in the 2016 data. So you can see that that deep, shallow, deep pattern was shifted about 25 days later in time when the ice broke out about 30 days later. This is really interesting from a biological perspective because, like I mentioned before, the ice breakout is what triggers the krill and fish spawning via photoperiod control. So it seems like maybe if the ice breaks out later, the shallow period may occur later because the krill and fish are spawning later as well. So if we take the shallowest date of diving for each of those years and plot them across the ice breakout dates for all 4 of our study years, we saw a really tight positive correlation between the shallowest seal diving date and the ice breakout date. There were also clear differences in the proportion of animals that became missing in the following year. So I'll show you what I mean by that by adding a right y-axis, a proportion of sexually mature females that became missing the following year. For 2015-2016, when the ice broke out very early and very late, respectively, about 20% of animals went missing. Whereas during 2013 and 14, when the ice breakout date was intermediate, between 5 and 10% of the animals went missing. So we still have a lot of work to do in terms of figuring out the mechanistic drivers of colony attendance, but I think by looking at the influences of ice dynamics on body condition, we may be able to better characterize those links. What's really interesting is that ice breakout also corresponded to the date of molt start. So again, in that really late ice breakout year, we saw that the molt was about 10 days later than it was during the other years. This relationship was less strong than that of behavior. But as I mentioned before, late ice breakout has been found to both delay and stunt the phytoplankton bloom, and so that can lead to changes in food availability. And food availability, in turn, with other species, has been found to influence molt phenology. So higher food availability has been found to advance molt onset, and lower food availability has been found to delay molt onset. And so I suggest that a similar mechanism may be acting in this system. Okay, so clearly there are many components of the molt that require future study. In general, the energetics of the seal molt are not completely understood. We know that thermoregulatory costs are a function of the difference in temperature between the environment and the skin of an animal. So for example, when animals are molting, their skin temperature has to increase in order for them to produce new fur. And so the differences between environmental temperature and skin temperature are increased, and therefore thermoregulatory costs are increased. We know that similarly, if they molt late or if they go in the water, their thermoregulatory costs could increase. So the effects of molt on activity and the energetic repercussions of that are part of Skylar Walcott's master's thesis that she's working on. Similarly, the interactions between hormones, body condition, and the timing of molt are topics of current study in Jen's lab by Amy Kirkham. So one way to obtain sequential mass measurements during the molting period, both to understand the energetics of molt, but also just to understand the annual mass dynamics of these seals, is through photogrammetry, which is the measurement of mass and volume in photos. So for one of the chapters of my dissertation, I evaluated the accuracy of several morphometric and photogrammetric techniques for estimating mass in Weddell seals. I found that most of the commonly used techniques did produce mass estimates that were not significantly different than actual mass. And for the one metric that didn't, I provided a correction equation so that it can be used to estimate mass in these animals. So I don't have time to talk about this chapter, but I'm happy to answer any questions that you have at the end of the talk. Okay, so I want to summarize what I talked about today. I found that molt is an important component of the annual cycle. I found that life history events are tightly linked. So specifically, successful pupping leads to both increased foraging effort and later molt timing. And later molt timing can influence colony attendance in the following year. And I found that these life history events are modified by ice breakout timing. So if we put all this information into sort of a holistic framework, we knew before that successful feeding leads to increased blubber stores, which can be used in these pinnipeds for thermoregulation and storage, and therefore enable survival and reproductive success. But we now have more information about seasonal prey distributions and how they change across the summer based on the data from our Weddell seals. We now know that reproductive seals make more dives and that they molt later. And then we know that molt phenology then influences colony attendance. So now that we know that these life history events are linked, the $1 million question becomes, how can we sort of predict how these animals will respond to global change? And I think that by looking at the ecology coupled with the behavior and the physiology and the evolutionary drivers of these life history strategies, we can sort of better understand the fundamental components and better be able to predict how they might change in the future. I want to conclude by just spending a couple minutes talking about the outreach activities that I've taken part of, which allowed me to both become a better researcher and become a better science communicator. Antarctica is a really special place, and so when we returned from the ice for the first time, Amy and I worked with our outreach coordinator to start a classroom visitation program in Anchorage and Fairbanks. So we sewed this stuffed seal that we took to the Antarctic, and we took to classrooms all over Alaska. And at each of the classrooms, we would talk to students about the size of the seals, allow them to measure the seals, talk about their anatomy and their behavior. And then each of the students wrote their names on one of the colorful patches, and the seal became known as Patches the Seal. So I had a second grader come up to me and ask if I would write a book about Patches the Seal. And so I worked with my fiance to write this book that's now available on Amazon. And I'm happy to say that It has sold out of its first print, and we're in the process of using the very small amount of proceeds that we get to translate the book into Spanish and to donate it to underserved California communities. So it's really exciting to know that thousands of these children are able to sort of metaphorically put themselves in our shoes and see what it's like to become the Antarctic explorer. So with that, I want to thank the huge number of people who've made this dissertation possible, to the National Science Foundation and National Geographic for funding the work, again to my committee for making this all possible, especially to Greg for allowing me to be a student here and being really supportive, and to Jen for allowing me to be part of the life-changing field research. To my collaborators and supporters at UCSC and at UAF, especially Joe and Matt, who listened to this talk yesterday, and to my cohort for welcoming me here. And finally, I just want to give a personal thanks to, not gonna cry, to the field team that was there throughout this whole process, and to my friends who got me healthy and happy and running, and my family for supporting my crazy dreams. And thank you guys for being here. They get really itchy when they start molting. Okay, so I guess I'll take any questions. Can someone let that person in, maybe? No questions. Yeah.
Excellent summary. Incredible project. So I'm really interested about your skippers. So if I'm interpreting this correctly, your skippers have a higher probability of molting early, and then those animals that molt early have a higher probability of becoming a skipper again next year, or is it go missing?
It's to go missing, yeah. So it doesn't appear to influence reproductive success. It does appear to influence colony attendance. But we know that both animals that are appearing at the colonies as skippers and animals that are going missing, neither of them produce pups. So it doesn't appear to have an effect on reproductive success, yeah.
So does that mean that being a skipper is kind of a portend for, not being in good body condition, and so you're just falling off the edge? Or is that treading on where Amy's going?
That's Amy's PhD. No, it's a really interesting question, because when the skippers show up at the pupping colonies as skips, they're really fat. They're in really good body condition. And so we're kind of trying to use body composition metrics to better understand how their mass fluctuates throughout the year. But it doesn't appear, the skippers don't appear to be in poor body condition. We're not sure that's why they're skipping in the first place. But trying to figure out why the animals are skipping or what the energetic benefits of colony attendance versus temporary emigration are currently being worked on. Yeah, there's another one. Yeah.
So you started off with talking about the timing of reproduction. I'm kind of curious if you follow that loop through. What do you think dictates when pupping is occurring? What are the mechanisms?
Yeah, it's interesting. So in Weddell seals, pup birth timing tends to be really consistent within individuals, and also the window is very short. And so I didn't actually find, I looked, but did not find links between molt timing in 1 year, so in January and February, and then pupping timing in the following year. And I think there are a couple reasons for that. The molt timing window is very broad, so there's a range of maybe 80 or 90 days across these reproductive categories. And I think that some of that variability is due to metrics that we aren't actually calculating, like body condition during the molt. But I also think that so much happens over the winter. They can gain so much mass. These animals are foraging 70 to 80% of the time during the winter. So they're working really hard. They're gaining a bunch of their body mass back. And it's possible that, for example, foraging success during the winter is better in some animals than others, or that timing of breeding, which we can't necessarily measure with high accuracy, are different across those animals. So I think there's a lot of variability that just we're not able to explain, and I think that's probably part of the reason that I can't actually go from pupping timing to molt timing to pup timing again. It's one link too far, I think. Yeah, that's the question. Yeah.
Great presentation, Roxanne.
Thank you.
I'm curious, is there any evidence or opportunity to even evaluate whether the skippers had been pregnant and have been
of our current research. So I didn't have time to talk about actually most of the research that we did in the field, but the project is based around an NSF grant that Jen Burns and Ward Testa got that supports a couple graduate students. And one of the main questions is, what are the physiological drivers of the different life history events? And so we actually handled animals by sedating them both during that pupping period and during the molting period by recapturing animals across the 2 seasons and across the 4 years of study. We did that for almost a lot of animals, many animals. And so we're starting to get much better insights into their pregnancies. And one of the things that postdoc Michelle Shiro was working on was detecting pregnancies when they're very small, on the order of centimeters. She was doing transrectal ultrasounds during the molting period in order both to figure out whether there's a period of delayed implantation or sort of when they're getting pregnant or when breeding might be happening. And so hopefully she'll come out with a couple of papers in the next couple of years. But to give you a sneak peek, I don't know if this is fair, but a lot of them get pregnant and lose those pregnancies. So it doesn't appear to be a pregnancy thing, it appears to be carrying those animals all the way through to the lactation period. Yeah, Brian?
In your reading about how fur and feathers and blubber act as insulators, did you ever find any comparative data that would show thermal properties amongst those 3, and particularly interested in blubber? Is it a good, does it retard heat flow as well as feathers and fur?
Yeah, so I think that the really part of that, did everyone hear the questions or not repeating questions? Yeah. The really interesting question there, I think, is how environment drives how good insulators are. So seals that spend a lot of time in air tend to rely on fur, like the otariids, like fur seals, they tend to have a lot of fur, and that fur can trap air, and it's really good insulation in air. But for example, the seals that are spending a ton of time during the winter underwater, fur is not as good of an insulator underwater as it is in air. And so they tend to be more of the capital breeders, more of the animals that have, like these animals do many centimeters of blubber on them, and they use fur more for like wound protection and maybe hydrodynamics. And so I think in general, in the water, blubber is a much superior insulator. In the air, it tends to be feathers and fur. And we see the influence of that in the molting strategies as well, right? So I think that's one of the reasons that seals and sea lions don't molt many times per year, because they don't rely on that fur for thermoregulation. They rely on blubber. And so while they do need to replace the fur at some point, it doesn't need to be multiple times per year for camouflage or multiple times per year for insulation. Yeah, does that answer your question?
I was wondering where movement dynamics and birds played into molting strategies.
Did you say movement dynamic?
Movements, like are they migratory, are they not? Does that play into your molt schematic?
Yeah, in many convoluted ways. So I originally thought that maybe migration would play, a really key role in driving the molt strategies. It tends to be not necessarily whether or not they migrate, but how far they migrate. So animals that migrate more often are faced with different environmental conditions in the 2 species that we're going. That's one of the, I think, key components that's being studied right now in bird dynamics, because a lot of species, molt strategies have been sort of described at the fundamental level. It's actually looking at how molt timing is integrated into the annual cycle. So if animals are spending a lot of time migrating, they might not have as much time to molt. So they can either do molt migration overlap or molt breeding overlap, and that has all sorts of energetic implications. Yeah, super interesting. Yeah.
Just to follow up on the molt strategies, did you find much information on tropical species, particularly tropical mammals, what they're doing?
Mammals in general were really, really poorly represented in the literature. I really struggled with mammals. I think that part of that is because it's really difficult to tell when they're actually molting. Like obviously I showed the pictures of the Waddell seals. It's really obvious to tell if they're molting or not or how far along they are, but with something like, a bear, it's a lot more difficult. And so the methods for actually figuring out when animals are molting are much different than in seals. Like there will be hair snare traps or, people do visual surveys. And so I think because of that, the molting literature for mammals is predominated by Arctic species where you can actually see the color change. Like snowshoe hares, it's really easy to document when they've molted or not and whether there's survival implications to that. I found almost no information about tropical mammals. I think seals and polar mammals were the predominant ones. But there's maybe molt descriptions for 40 total mammal species or something like that. It's really not that many. Birds much more so. I think both because they're more obvious, but also because the feathers are really important for them. Yeah, right.
So you care to speculate with an infinite amount of time, money, and people?
Whoa!
Then you could do comparative Waddell study in the various areas that are not like Waddell, and then add on the 3 other Antarctic species and see. And again, this is a lifetime and an infinite amount of money. What would you speculate at this point on the difference might be between these and crab eaters?
In terms of what metric? Anything?
Anything.
Dream
Just,
big.
yeah, just go big. Yeah.
Whoa, okay. Oh my gosh. Great. I'll tell you my dream.
Okay.
So the deep shallow deep pattern is really interesting to me. And it's just, there's such a fundamental lack of data pertaining to where fishes are in the water column, how that changes seasonally, and how that might drive both, top down and bottom up. So my dream is to put lots of jaw accelerometers on seals all around the continent to figure out if this phytoplankton shallowing period that I'm suggesting is in fact happening. If we can confirm that, that would be great. If we can figure out whether it's happening all around the continent, that would be really interesting. Just because the ice dynamics are quite different across the different stations, and across the different sectors of Antarctica. And I would pair that seal tags with underwater sensors that do acoustic Doppler profiling of the prey species. Because I think a lot of the prey information, the fish information, in terms of surveys are done from boats, which are great, but they move both in space and time. And so it's really difficult to disentangle these seasonal vertical distribution shifts. So that would probably be my dream, would be to put like thousands of moorings around the Antarctic that measure prey abundance in the water column throughout the different seasons, maybe coupled with phytoplankton concentrations.
Hey, Roxanne.
Hi, Ward.
I had a question. You made a comment about sort of the prevalence of social selection factors. I'm presuming you mean like sexual selection factors in birds. That it was more prevalent in birds than in mammals.
Yep.
So was that strictly about pelage? Because I was thinking sexual size dimorphism is probably the opposite.
Oh, yes. No, sorry. That was completely limited to pelage characteristics. Size is a very...
Like coloration.
Yeah. So if you think about, like lion's manes, obviously, are one example that you can think of where, like, larger manes are associated with mating opportunities. But it's not coloration for mate choice dynamics. So like in some of the baboons, like skin coloration can be used, but because it's skin pigments rather than hair pigments, it's disentangled from the molt, and so it doesn't contribute to driving the molt strategies of those species. Does that make sense?
Mm-hmm. Sure.
Okay, thanks.
And I was going to make just one comment too. You also were mentioning the effect of sea ice extent and the distribution of immatures, and that you thought that was primarily related to productivity and foraging opportunities being more restricted during extensive ice years.
Yeah.
And I read that Ian Sterling talked about that a little bit, and it brought up the fact that for immatures, the teeth are not fully developed yet, and Weddell seals use their teeth to keep breathing holes open.
Yeah.
So with the immatures, they're probably not as capable of maintaining breathing holes. So that may be another reason that they are displaced from or further from the colonies during extensive ice years.
Yeah, that's a really good point. So one thing I didn't talk about was the reason that one of the reasons that Weddell seals are the most southern breeding mammals, because they have really strong teeth and really flexible jaws, and so they can ream away the ice like this, and so they can maintain these breeding holes throughout the year. Whereas something like a minke whale or an orca just can't do that, so they can't get to areas that are as far south. So that I think brings about really interesting predator dynamics, right? Because actually in these animals, the colonies are safer than the areas that they would temporarily emigrate to. Whereas, for example, for a lot of bird species, I think probably colonies are less safe because that's where the predators go. So yeah, Ward, that's a really interesting point. Thanks for pointing that out. Do you know when their teeth are fully developed?
No, it really, I mean, it has to do with how fast the dentin fills in. And
Yeah.
so most of that is pretty simple by the time they reach sexual maturity, probably around 6 or 7 years.
Ah, okay. Cool, thank you. Yeah.
So I'll follow up on Ward's thing about pelage and size and everything. One thing you didn't mention here is that in this species, you can't tell looking at the distance.
Yeah.
Who's male, who's female, and they're all the same color. So the same color and the same size.
Yeah.
So do you want to speculate on which males are more successful?
Yeah, that's a good question. It's a funny system. Like Greg talked about, I came from the elephant seal world. And obviously, if you've seen an elephant seal before, the males are like 4 times larger than the females. They're like, what, 2 tons? Whereas in Weddell seals, males and females look exactly the same. So when we're doing our molt surveys, we can't tell if it's a male or a female until it rolls over and we actually see female opening. So I think that's, comes about for a variety of reasons. But I think one of the interesting things about Weddell seals is that they breed underwater. They have this cool mating system where the males sort of defend these underwater territories so they don't allow access to the breeding holes, or they kind of restrict access to the breeding holes during the breeding season. So typically during the breeding season, we see the males that are hauled out are either really, they've been attacked by other males, and so they're like bleeding from the flippers and in kind of poor shape. The males we don't tend to see as much during the breeding season are the ones that are underwater actively defending territories. And so one thing, I like that you bring this up, because one thing I really want to do, I have all of these molt timing data for the males. It would be really cool, I think, to look at when, there's quite a bit of variability, like 60 days or something like that, in molt start dates across all of the males. And so it'd be really interesting to see whether males that spend more time underwater defending territories kind of put off the molt until later, and whether the juveniles or the ones that are not quite as successful molt earlier. And I think that could help address one of the big questions with the molt, which is exactly how much percent time foraging plays into when the molt occurs. So we know from heat flux data that the seals lose a lot more heat to water, maybe a few times more than in air. And so if they're losing a lot more heat underwater, they don't want to molt in the water. So that can maybe give us some insight into sort of the energetic implications of spending more time in the water. Yeah.
Any more questions? All right, let's thank Roxanne one more time. Yeah, so again, there's a potluck tonight at my house. And if you want any, you can walk there from here, 1756 Reed Circle.
No, I'm tired.
And I can give you, if you want a map, I'll just let you know and I'll send it on to you. So we'll start around 6 tonight and go 9 or 10.
Thank you guys for coming.
I need to write that letter. I'll take it down there and that was awesome. That was good job.
Word, are you still here?
I'll be back. Text me.
Can I just hang out?
It's going to let it wet.